Okay, everyone, let's get started. So this is the first try at some of the lectures we might do. This might be the only video you see from me going over a brief lecture portion that we need to cover in this class, or it might just be the start of the final material. We'll see how this pans out this week. But let's just start in. So this is the renal physiology lecture. A lot of what we're going to talk about today is just understanding the basics of kidney anatomy and then tying a little bit of how the structure of the kidney relates to its function and the specific cell types you're going to see in the kidney. So here are the study objectives I would know if I were in this class, right? So understand the basics of the kidney. This would be great for those of you who haven't taken anatomy. Understand how fluid is processed. So what we mean by this is what pathway does fluid follow through the kidney? Like you need to know the glomerulus and Bowman's capsule and then all the tubules and whatnot. So we'll get through to that. What affects GFR or glomerular filtration rate? Sorry about that. So what factors are we going to be looking at? Just a little preview, you're going to be looking at the different pressures in the arterial, hormone levels, and autoregulation of the kidney. So we're going to learn specifically about filtration, but we'll learn about the other functions that the kidney can do, including reabsorption, secretion, and excretion. But we'll go a little bit more into depth in later lectures about how we regulate these three things. These are just a bunch of good terms that you'll need to know, and by the end of this lecture, I hope you can flip back to these study objectives and be just very familiar with these terms, and we'll go over the cells. So let's get into it. So structure of the kidney, first things first. So kidney is the bean-shaped organ. It sits retroperitoneally, that's not the right word. It sits in the retroperitoneum, so that means just kind of in the back of your body, so that's why someone kicks you or punches you really hard in the back, you can have kidney issues. So if we look over here, this is a kidney that's cut in half, or the cross section of the kidney. You see all of these little, let me get it back here, sorry. You see all these pyramids, right? These pink tissues that are surrounded by these blood vessels are pyramids. And that's traditionally considered a part of the medulla of the kidney. And remember, just when we say medulla of an organ, that's the interior portion, or what's away from the surface. And then out here, all along the edge, we see the cortex, okay? So cortex on the outside, medulla on the inside. The kidney is one of the most important organs. You need to know the difference between the cortex and the medulla. I'd say the most important is the adrenal glands, and because they secrete specific hormones. You don't need to know that for this lecture. But this is the kidney in general, but we need to learn more about the functional units of the kidney. And we're going to get into that in just a moment here. Here's the side view. It's going to be the nephron is what's going to be our key regulator of what happens in the kidney. So kidney anatomy, bean-shaped organ functions include all of these nonsense, and we'll go over those in the lecture. So if you don't know anything about that, that's totally fine. But here's another view, right? So it's kind of the cross section of up here. This is the cortex. This is the medulla. So interior of the kidney, exterior of the kidney. And you see these tube-like structures, right? You can think of these as the precursors to the urethra and then the ureter. So this right here is a collecting duct. This is all the tubes we'll have to learn. Anyway, when you're looking at this thing in general, this is called a nephron. And let's get into that next slide here. So the nephron is the functional unit of the kidney. So it goes all the way from the glomerulus to the proximal convoluted tubule. And we just say that because look at this tube, right? It's very convoluted. So that's kind of an easy vocab term. It's the proximal convoluted tubule. Okay, good. Now let's move down. So now we have the loop of Henle. And we call it the loop of Henle just because it's a big loop, okay? So the kidney is named after some pretty distinct structures. And please don't think, oh, this is so dumb, I have to know all these dumb structures. Each of these structures does something very specific that has been tested, that we know about, that you will be expected to know for the final in this class. So you have the descending loop right here of Henle, the ascending loop, distal convoluted tubule, and the collecting duct. Each of them do have their separate functions, so that's why they're listed here. You don't need to know them now, but we'll get into them in future lectures and a little bit into this lecture, okay? So we already talked about this. This is just kind of a recap, but what we need to show you here, so let me go back a slide real quick here. So this is the nephron. It's folded out, so you can actually picture the nephron like this to help you learn it better, but just realize that in real life, the nephron is going to be all folded up on itself like this. And what's really cool is that you actually have capillaries following the pathway of this loop of Henle all the way through all these tubules so that there's exchange between your blood and what's being excreted into these tubes. So anything that comes into these tubes is on a path to be excreted in your urine. Now, it doesn't have to stay in these tubes. It can get back into the capillaries through the process of reabsorption, which we'll briefly touch on in a minute. But just know that if you see anything in purple, which then leads to yellow over here, this is what's going to come out of you. And then what's in red can then bring back solutes, it can bring back water and a bunch of other stuff into your system if your body needs to keep it. Your body regulates the kidney function very tightly to control what you have. So one last term we need to know here is the difference between the cortical and the juxtamedullary nephrons. So the cortical nephrons, this terminology you need to know is just about where the nephrons are located. Cortical nephrons, which make about 80% of the nephrons in your body, look like this guy right here. So the nephron, if it goes down in the medulla, it barely goes down into the medulla. It's primarily found in the cortex. However, sometimes you have big guys like this that dip all the way down into the medulla, come back up. This is the juxtamedullary nephron. So you see juxtamedullary nephron, you think, oh, that's really long nephron. What's actually kind of cool is if you learn, and if you guys ever have the amazing opportunity like I did to dissect a camel, just kidding, that never happened. But if you dissect a camel and you could look at their tissue and see their nephrons are actually all very, very long. And what do we usually associate camels with? Well, that would be the capability of storing water for a long period of time. And the very, very long loop of Henle's that dip very low into the medulla allow them to have that ability, which is kind of cool. Okay, this is just a preview. We'll talk more about this at the end of the lecture when we talk about the autoregulation of the kidney. But the juxtaglomerular apparatus is this right here. Let me circle that a couple times just so you can see it. Actually, I can just circle it with a pen here. 1 second. So this is the portion you need to know. Oh, that green's not showing up well. Sorry, guys. Anyway, so what's happening here is you have a couple different cell types. You have the afferent arteriole and the efferent arteriole, and then the distal convoluted tubule actually comes up. So we saw in the nephron that we laid it out kind of flat in that slide. You can picture the nephron moving with the fluid moving left to right. In real life, like we said, it bounces back, like it comes back over. So this is the ascending loop of Henle. I'm sorry, I said that was the distal convoluted tubule. That's my bad. The ascending loop of Henle has these specialized cells called the macula densa cells. We'll learn about their function towards the end of this lecture. Then we also have the juxtaglomerular cells right here that sit on the afferent arteriole. So let's go over the blood flow into the Bowman's capsule while we're here. So picture having a really big artery right here, not a really big artery, but a bigger artery that flows right here, and then the blood can flow into the afferent arteriole. Okay, so once blood flows into the afferent arteriole, sorry, let me actually change that to red, like blood. So flows into here, comes into what's known as the glomerulus, right, which has all these crazy looking cells on it. The glomerulus is just this semicircular big mesh of capillaries that sit inside this capsule. You see this yellow capsule here, that's Bowman's capsule. So this blood will flow through these capillaries, and you have, we'll talk about in the next slide or two, the specific capillary type you have here that allows for fluid exchange between what's inside the endothelial layer, and this would be the endothelial cells, right, that are all throughout here, allows fluid to move through the endothelial layer into Bowman's capsule. Once it's in Bowman's capsule, it starts going down the tubular system into the nephron like we talked about. So that's the afferent arteriole bringing blood into the kidney, not into the kidney, I'm so sorry, into the JGA is also what it's called, so I'm going to highlight that, but we're talking about the juxtaglomerular apparatus. So afferent arteriole brings blood into the JGA. The efferent arteriole brings blood away, and then this efferent arteriole, like we saw in the nephron slide, actually will form a capillary network that surrounds all of the tubes of this nephron that's going to be happening. So those are what just in general, the cells you need to know about the JGA. We'll see those a little bit later on and their specific functions. So let's keep going. So the main thing we think of when we think of the kidney is its ability to process fluids, meaning it can excrete urine, which is primarily made of water, any plasma proteins, drugs that are in our system. A lot of them get excreted through the kidney. So how does this all happen? There's 4 terms you need to know here, and I would know these pretty well because they all mean something very specific. So filtration is just the passive flow, right? There's no active transport here. The passive flow of solutes, liquid, which that liquid would be water, into Bowman's capsule. That's the initial filtration that the kidney does. So the majority of what's going to be excreted in the end will actually be dumped out here in the Bowman's capsule. Then you see all these R arrows right here. That means reabsorption, meaning whatever was put into the kidney, whatever portion of the tubule, right, it comes back out, and now it's in the capillary. And as you can see, the capillary follows the lumen of what's inside the nephron so that you can have exchange. Secretion, so you first filter something. Sorry, guys, let's go through this from the beginning. You filter something out. So say we have a little molecule of glucose that gets filtered out. No, let's make it the drug penicillin. So penicillin gets filtered out here. It gets reabsorbed, okay, so it can make it back into the bloodstream, but then we can secrete it back into the nephron because we might not want to keep that. So secretion can be active transport, and there can be cases of non-active transport. And when we say active transport, we're meaning ATP or energy driven, right? So that's the first, those are the main 3 you need to know. And then excretion's kind of easy. It's just lumen out to the environment. Something that leaves the collecting duct will get excreted. So the collecting ducts all come together to form the drainage system that forms the ureter, right, that then goes into the bladder. So those are the 4 functions of the kidney that I would be very familiar with if I were you guys. If we said, hey, where does filtration occur? Or like, where are the locations? You would say, oh, filtration can only occur here at Bowman's capsule. Where can reabsorption occur? That can actually occur in nearly everywhere, okay? So know the differences. And this is really good to know because there are specific sites. For example, right here, the reabsorption of the descending loop of Henle is very sensitive to water. And over here, it's very sensitive to solutes. We'll get into that. Please don't memorize that at all. Just realize that these arrows are not all equal in what's being reabsorbed and what's being secreted, okay? So solute processing, this is the equation that I would know. So amount filtered minus the amount reabsorbed plus the amount secreted equals what's excreted. Okay, so it's pretty simple. I want you guys to be comfortable if you had to do a calculation like this at any point in the class. You take the amount filtered. So say that is 100 milligrams of sodium I filter out. I'm getting rid of that, it comes out. And I reabsorb 10 grams of sodium. So, okay, so now I only have 90 grams here. Okay, that's fine. But now I secrete 20 milligrams of sodium back. So now I actually have 90 plus 20. So I have 110 that are going to be excreted, even though initially only 100 was filtered. So that's just an example. That's not necessarily how sodium functions, but I hope that helps you see if reabsorption is larger and so secretion is smaller, you can have different events from filtration to excretion, okay? So filtration, just the basics, the very first step. We're going to go a little bit more in depth into that. So you filter directly into Bowman's capsule. So coming in through the afferent arteriole, makes its way through this capillary bed. And I ignored these cells earlier, but it's time to talk about them. These cells are called podocytes. And this is an electron micrograph of podocytes. They look kind of crazy, right? So you've got like all these little fingers that come together on these cells to close the gaps in the capillaries until they specifically signal what they want to be released. So let's go into the next slide because that helps us see it a little bit more. So this is just this slide right here. Let me go back just so I can show you. Is just equivalent to the cross section here. So the next slide we're going to look at is smaller in proportion, just a single part of the glomerulus and Bowman's capsule, okay? So right here, we have the capillary and we have the endothelial cells. And there's something very important that we need to know here. This is a fenestrated capillary. Let's see, is that written anywhere? No. Okay, let me write that out for you guys. And this is going to be really bad because I'm trying to write with like my pad. No judgment, please. Okay, I would be familiar with that term. If you can read it, that is fenestrated capillaries. And what that means is that you actually have slits in between each one of those little endothelial cells. So let me highlight that here for you. Okay, so right here, this is an endothelial cell, meaning it's on the blood side. This is in the arterial. There's actual holes in between the cells. Now, we can get stuff to come through. Now, these holes aren't big enough that red blood cells can come through. So when someone starts peeing blood, we know that's a really big problem because something bad has either happened here or further on in the process. We do not want any red blood cells to get through these holes. Now, it is big enough, however, to get proteins, can get drugs, it can get molecules like glucose through, and then it can try and get to the lumen. So there's 3 layers here. Endothelial, that's the first layer you should know, okay? Endothelial layer. Second layer is what's called the basal lamina, as seen right here. This is the basal lamina. And what this is made of different glycoproteins, such as, actually, I'm not sure exactly what the glycoproteins are. I'd have to look that up for you guys, but it is a bunch of negatively charged glycoproteins. And when we say glycoproteins, that just means proteins that have sugar molecules attached to them that form kind of this barrier that they have to get to. If you guys know what a Western blot is, I think of it as that. Like it has to go through these little tunnels. So a protein here gets through this gap, then it has to get through here. And what's really cool about this basal lamina is that it's actually negatively charged. So negatively charged proteins won't try and get, they'll be repelled. So if they do make it over here, the basal lamina will push them back in. Whereas other solutes such as water, maybe glucose, they can still make it through. Then the last layer here is the foot of the podocyte. So let's look at the podocyte over here, this big guy right here. So on the left hand, we have this podocyte that reaches out and he covers the slits of the endothelial cells. Now, when he's signaled, he can open up holes so that stuff can get all the way out through the podos, through a slit over on this side. So really, there's nothing here. If there were no podocytes, you would have no barrier and everything's just flowing out at all times. The podocyte really allows there to be a specific secondary structure that's blocking transfer. Last cells we need to know are the mesangial cells. Those are just in the middle. These cells can contract and they also really help to keep the kidneys clean. They're not the most important cells to know, but I would know that they are very prevalent in the nephron and that they are important in contraction along with the podocytes. Okay. Oh man, that's really ugly. I'm sorry, guys. Fenestrated capillaries. That just means capillaries with holes. Okay, reabsorption is really high. And what I like to think of that helps me remember this is the molecule glucose. So glucose actually does get filtered. And you may be thinking, what the heck? Why would glucose ever want to get filtered out? Our bodies need as much glucose as we can possibly have. Well, we can't really control all that much what gets filtered. Stuff is going to get out if it gets out. Now, not everything is filtered, right? So about 20% of renal plasma flow is filtered. This is a number I would know, okay? Can't guarantee that's on a test, but I would generally know that the majority of blood is not necessarily filtered like we think it is, so only about 20%. So let's follow a little molecule of glucose. I'm a molecule of glucose coming in here through the afferent arteriole. I get into the glomerulus and I get filtered. Okay, now I'm going to be excreted if nothing happens. What's super cool in patients who do not have diabetes mellitus, 100% of the glucose is reabsorbed back into the capillaries. So when you think of diabetes mellitus, right, you think of, well, the urine is sweet. So what's happening? There's so much glucose that's getting filtered because the blood glucose level is so high that the reabsorption transport molecules cannot get all of the glucose back in. And now we have a problem. But in general, just remember that reabsorption is very high. Like the body can keep what it wants to keep, even though it gets filtered out. Okay, so that's my example. Just think of a little molecule of glucose coming through. He's going to get reabsorbed 99.99% of the time if a patient doesn't have diabetes mellitus. If they do, it won't be efficiently reabsorbed and it'll actually be excreted, okay? So calculating GFR, remember GFR is glomerular filtration rate. Super important to know that term. So what we're looking at is a bunch of different pressures. So when you think of Starling's forces, do you guys remember where you last saw this? It was in the heart and understanding specifically the pressures that govern the heart. So here, let's start here. So we have the hydrostatic pressure or the blood flow pressure. That's what I like to think of as. The pH is about 55 milligrams of mercury. So that pressure is created by blood simply just flowing here all throughout the glomerulus, right? Because it's in an artery, so it's still decently quick. Not an artery, it's an arterial, but still decently quick. And you have about 55 milligrams of mercury here. Okay, now what's this little pi guy? I don't hope you guys would remember this from the capillary lectures, but this is actually going to be the osmotic colloid pressure that pushes back into the glomerulus, right? So there are fluids out here that are pushing back, trying to get back in, and that would be annotated by the P fluid here. So I would know these three separate pressures: the pressure from the blood pushing out, pressure from fluids pushing in, and the colloid osmotic pressure that can be from proteins or other solutes that are trying to push back in. But in general, we usually have a net filtration pressure out of the glomerulus of about 10 milligrams of mercury, okay? So that's enough to actually get a decent amount of filtration through all those fenestrated capillaries. These are some questions you can ask possibly during class when we have a little bit more time to go over these questions. This is one of the coolest things about the kidney in my mind. So there is significant autoregulation of the kidney, meaning the kidney can keep its glomerular filtration rate, or GFR, at approximately constant value all by itself. So here we have the mean arterial blood pressure. So it's all the way down here at 40. This guy's might be dead. And then you can get all the way up to 200. So these pressures can change dramatically, right? If I'm working out, all of a sudden my mean arterial pressure is going to skyrocket, but my GFR is going to stay the same. Think about how bad that would be. If your mean arterial blood pressure translated into GFR, you might blow up your kidney because there's so much pressure. So it's amazing that the kidney can do this. We have to learn specifically how the kidney does this, okay? And we just talked about those things. So the two mechanisms, the myogenic response, I think in my mind, the myogenic response is a little bit easier to understand. And then we have number two, the tubuloglomerular feedback. This one's a little bit more involved. We have to go back to the slide of about the JGA, or the juxtaglomerular apparatus, and understand how the macula densa cells are talking to the afferent arteriole. So we're going to start with the myogenic response to try and help this, okay? Help us understand how the kidney autoregulates its GFR. So arterial resistance effects. This is a great image from your book, and I think it's very helpful. So let me pull up all the info here. Sorry, guys. Okay, so let's start right here. So this is like a clamp. Pretend that we can put a little clamp. We can either tighten it down or we can loosen it up like we will here. And what effect will that have? So first of all, if we tighten this, let's not tighten this down. Let's just keep it normal like it's not doing anything. So the arterial blood flow, or RBF, renal blood flow, into the glomerulus is going to stay the same. GFR is going to be not increased per se, but this is just our baseline, right? So it's at a certain level and our GFR is at a certain level. Okay, now let's say we constrict the afferent arteriole. In this case, we're using a little clamp, but our kidney can actually do this, and we'll learn about how it does that in just a second. We use a little clamp, not as much blood flow is getting in here. And what does that mean? That means that the pressure of this blood is lower because we're not getting as much blood pushing up against the vessels of the glomerulus, which will then decrease the GFR. Okay, so that's the first thing, right? If we vasoconstrict the afferent arteriole, we decrease GFR. Now, how about what happens if we increase, I'm so sorry, if we constrict the efferent arteriole. Well, this is still going to decrease the renal blood flow, right? Because it can't get out as quickly. So it's probably going to either slow down or something's going to happen here so that there is less blood coming in because less blood's going out, and the blood is not going into Bowman's capsule because of the tight fenestrations in those capillaries. So we actually get an increase in GFR here. And why is that? Well, if the blood can't escape, it's actually causing an increase in pressure. More blood is getting pressed up against these walls. Okay, so think to yourself, what's going to happen now if I decrease resistance? So, and comparatively to here, decrease resistance in the afferent arteriole. What's going to happen to renal blood flow, GFR, and the hydrostatic pressure of blood? Just take a couple seconds to answer that, you guys. You can pause the video if you want to think about it for a second. Okay, let's go over it. So if I decrease the constriction here, that also means that I am going to be decreasing the resistance. Sorry, I blinked there, which allows more blood to come in. That's going to increase the pressure, more blood coming in, more blood pushing against the walls of the glomerulus, increase the pressure, increase the glomerular filtration rate. Okay, now if you understand this, you're ready to understand the myogenic response. So increased pressure in the afferent arteriole, maybe caused by something like this over here, activates stretch channels in the cells. Those stretch channels allow for a depolarization of the membrane, just like we were talking about with all the other Nernst-related membrane potentials. Those stretch channels allow depolarization. That leads to calcium coming in, and the calcium then causes constriction in the smooth muscle cells surrounding the arteriole, which is amazing, right? So we learned about how calcium causes constriction in smooth muscles in earlier lectures. This is the first form of autoregulation. If the blood pressure stretches out the walls of the afferent arteriole enough, the afferent arteriole will constrict itself. It's absolutely amazing. It can do that without any input from the body. Okay, so remember this, that now we're going to get into step 2 of autoregulation and how that happens. So I want you guys to just take a second to remember this whole apparatus here. Okay, so we have the juxtaglomerular cells in red, we have the macula densa cells in yellow, and we have this whole structure forming here. So let's go over it. Don't pay attention to this yet. We'll get into that in the next slide. So tubular glomerular feedback. Let's just go step by step. So we have an increase in GFR, okay? That increase will then, obviously if we increase the GFR, so more stuff is getting filtered out, that's all that the GFR is telling us. We're going to also increase the flow through these tubules, right? So more filtration means more flow. Okay, that will then flow past the macula densa cells. Let me go back a slide. Remember, these macula densa cells are specialized cells found in the ascending loop of Henle, okay? So what happens here is more liquid is flowing past these cells. These cells can then signal to either the afferent arteriole itself for a certain response, or the JG cells, the juxtaglomerular cells, to cause a response in the body. So flow past macula densa increases. Paracrine, and remember, paracrine signaling is just signaling from cells very close to one another. From macula densa to afferent arteriole, you cause constriction, resistance goes up, pressure goes down, GFR goes down. If you don't understand these last 3 steps, go back a couple minutes in the video and go over those little pictures we just did a couple slides back. These are just some great things to know. I'm not going to specifically talk about them. You can ask questions more during class or office hours if you have time. This slide is a lot of words, but it's very important to understand. So let me read this through. So when distal tubule flow is high, less sodium is reabsorbed, and sodium load specifically in the tubule is high. Okay, so you have more sodium in the tubule. If the flow is high, it doesn't have time to be reabsorbed. So the macula densa cells release adenosine. I should have put this in bold too. I would know what is released. The macula densa cells release adenosine, which causes the constriction of smooth muscle. So all of a sudden, sodium is passing past these cells. The macula densa release adenosine, specifically talking to the afferent arteriole right here. Sorry, not the efferent, the afferent arteriole here. That afferent arteriole will then constrict under the regulation of adenosine. So that's option number one. If the tube flow is too high, so we have a high GFR, a lot of liquid in the tube, we will release adenosine and we will target specifically the afferent arteriole for constriction. Option number two, when the distal tubular flow is too low, more sodium is reabsorbed, and now we have a lack of sodium in the tubule, okay? The macula densa cells release two things: nitric oxide, which we learned before is a potent vasodilator, and prostaglandin, okay? The nitric oxide causes vasodilation of the AA. Hopefully you guys know that's the afferent arteriole. The prostaglandin stimulates renin release from the granular cells, which are the juxtaglomerular cells. I think most times you're going to see them as juxtaglomerular cells, but be familiar with the term granular cells. They're one and the same, okay? Granular and the JG cells. And then that increases the mean arterial pressure and you get an increase in GFR. So option number two, low sodium, low flow past the macula densa cells. The macula densa cells are what is sensing this specifically, okay? They will signal release nitric oxide that goes directly to the afferent arteriole and tells it to dilate. And it releases prostaglandin, which tells the JG cells to release renin. And we'll learn about the renin cycle later on. Just know that renin is a great, great hormone that's super important in our bodies, and it helps increase blood pressure, okay? So I would be able to diagram exactly what's happening, draw a picture out, understand the differences between the myogenic response and the tubular response on the negative feedback about what's going on here, okay, you guys? It's super important to understand. This last slide, I think, is a little bit much for our level of understanding, even for mine, right? But this is just telling us specifically, not that I know more than you guys, that sounded kind of cocky, I didn't mean that, but this is telling us exactly what pathway is happening. I think you'll be safe if you know adenosine is important, nitric oxide is important, and calcium is important. Okay, guys, so that's the first lecture that we're gonna probably be posting on the YouTube channel if this is the pathway we choose to go down. I hope you learned a lot. Please tune in later for more information, rewatch these videos. If anything was confusing, please email me with any concerns you may have so that I can make these videos better for you. And please email me early once you watch this saying, Morgan, this part wasn't explained very well, or maybe you should go into depth here, add another slide here, so that the future lectures can be very beneficial to you. Okay, guys, thank you so much, and I hope this was great, and I'll see you hopefully in class.